This is episode 322 of the Stem Cell Podcast, Healing Hearts with Macrophages, with Dr. Filipa Simões. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The pod is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying this listen, rate us, leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on who we should welcome onto the show. Today, we are welcoming Dr. Filipa Simões, who's from the University of Oxford. And she's on the podcast to talk about her research on how macrophages interact with the cardiac environment to enable heart regeneration. We've also got our usual roundup of recent highlights and stem cell news coming up in just a sec.
But first, if you're looking for the best talks and networking in stem cell science, ISCR 2026 is the place to be. From clinician scientists to biotech founders to AI innovators to basic biologists, this meeting brings it all together. Discover the breakthroughs, technologies, and clinical insights you can take straight back to your lab. July 8th to 11th in Montreal. Register now at iscr2026.org. We're going to get started with something pretty relevant to our guest here today. It's very translationally oriented, and it's, I think, a big milestone in cardiac regenerative medicine, of course, something I pay pretty close attention to. We talk these days at the 20th anniversary of the derivation of iPSCs about how these things are rapidly progressing towards the clinic now. We've highlighted a number of clinical trials in various tissue types, from pluripotent stem cell derived tissues and iPS derived tissues, and so this is a big one. This is a phase 1/2 clinical trial result in the New England Journal of Medicine, one of the most premier clinical journals out there, focusing on iPS derived biologic ventricular assist tissue for heart failure, and this is work that's been led by Wolfram Zimmermann over in Germany, but also the BioVat heart failure investigators that are in different places around the world. The concept here is relatively simple. It's basically using a patch, almost like a Band-Aid, of iPS-derived cardiomyocytes and other engineered heart tissues, cells and then engineered heart tissues, to serve as a bridge in patients for heart failure. So this biologic ventricular assist tissue, or they call BioVat, is created from EHTs and engineered heart tissues, engineered heart muscles comprised of cardiomyocytes, stromal cells that are derived from important allogeneic iPSCs for cardiac remuscularization in patients with heart failure and a reduced left ventricular ejection fraction. And that means basically in that situation of heart failure, the left ventricle, which is the one that does most of the work, is not able to do a lot of work. And so the concept is simple. It's something that's been hunted for decades now. And maybe we're a little closer thanks to this particular study. And I've had a sort of a personal view into the development of this work because Wolfram Zimmermann is a pretty close collaborator with none other than Joseph Wu, my former graduate mentor and guest of the show here. I know Wolfram has consulted with Joe in years past on the best way to derive these iPS cardiomyocytes for therapeutic applications, and I think this is a long time coming. So these are the results in an AJM from an open-label phase 1/2 study of these tissue-engineered heart patches, and they were able to transplant these biovats into patients with heart failure. And these patients with heart failure and a ventricular ejection fraction of 35 or less, and at least one hypokinetic or dyskinetic left ventricular segment were the target population here, and they were treated with these BioVat allografts, which it's almost like a layered brick kind of approach here. It's like a layer of Band-Aids, which consisted of either 5, 10, or 20 of these engineered heart muscle units kind of stacked on top of each other. One other very important thing to note here is All these patients received immunosuppression. So it's not a perfect double-blind study, blah, blah, blah. There are things to consider here, but that is an important note. They, of course, looked at safety, assessed as adverse events related to the procedure, and they looked at different safety and some efficacy endpoints as well, including the heart thickness, the remuscularization, the target heart wall thickness, and improvement potentially in the left ventricular ejection fraction. Um, and so what did they see? There were, 20 patients. Again, phase 1, 2, pretty small scale here, predominantly focused on safety. 20 patients were treated in the study. 3 patients unfortunately died during the study. But apparently these were not related to the patches themselves. One was from vasoplegia, one actually from COVID, and another from aortic dissection. So again, not directly related to the transplant or to the procedure here. One other patient actually did undergo heart transplant, and in an additional 4 patients, actually immunosuppression was discontinued because of an implantation of an LVAD, a left ventricular assist device. There's some other side effects as well. So in the end, I think of the 16 patients who were treated with the safe maximal dose of these 20 engineered heart muscle units, 12 of them actually ended up completing the pre-specified 3-month follow-up. And in those patients, if you average it all out, there was a mean increase. There was an average increase in the target wall thickness of about 4.5 millimeters. Um, there's an improvement in injection fraction, modest, about 4%. Um, and an improvement in this thing that they call the Kansas City Cardiomyopathy Questionnaire overall summary score, kind of like how they're feeling. Um, they all had one adverse events. They straight up say all the patients had at least one adverse event, but sounds like it wasn't directly related to the patch itself. So again, it's a small scale study, but I think a really important one. One thing that didn't pop up here was arrhythmia, which I think is pretty exciting if you consider arrhythmia has perhaps been a major issue with these cardiac cell therapies. And I know Chuck Murray has done a lot of work in this, and we've talked to him about this as well. Um, but you get improvement in function, you get an improvement in the remuscularization of the heart, in the overall lifestyle of the patients. And I think this warrants a larger trial, and I think that's inevitably what they're going to be doing here. It's again phase 1, 2 trial. It's preliminary, but I think it's going in the right direction. What do you think?
I love the study. I have mixed feelings about what it portends for cell therapy in the heart. Uh, as you said, I mean, this is a motley crew. All the patients had at least one adverse event, and that's not at all unexpected, right? This is kind of this compassionate use cohort. There is no intervention here that's going to help these patients. This is uh, really for posterity on many levels. It's for us now. There's a lot to be learned, is what I'm trying to get at, and a lot was learned from this, as you said there. The lack of arrhythmia, I think, is a big takeaway here, showing that this kind of patch thing, unlike the injection of cells into the infarct or the muscle generally, where there's this potential for kind of, I don't know, a more uh, disordered uh, construction of the business end of that cell therapy. Yeah, I could see why this is preferential, and the lack of any arrhythmia, I think, is a really strong indication um, in that direction. The the other results that jumped out to me was like, yeah, the significant, like clear increase that you had in the target wall thickness, right? Very strong p-values, which seemed a little discordant, not necessarily discordant, but when you look at the degree of significance of like the actual functional improvements and the quality of life scoring, which are much more modest, as you said, I think that kind of discordance raises a question of like, all right, adding anatomical feature or architecture does not necessarily translate to a functional improvement, which is something that I think you intuitively know. but here makes it clear. And that's I guess my takeaway from this in terms of what it portends for cell therapy in the heart is like we're learning so much. And a big part of the takeaway for me of what we're learning is that it's really nuanced and complicated to integrate into the machinery of the heart, and particularly in a damaged, old, beat up organ, right? And I think on the flip side of that, in terms of the economy of the treatment, I wonder like how large scale do you go with the trial of this before the return is like, wow, you know what, this is just too expensive, too cumbersome. It's just not practical to do this to like start to meet the unmet need out there, right? The problem is just so big that I think that this, I don't want to use a pun, but like this band-aid kind of approach, it's just not practical at the current state of the art. That said, like you gotta have increments of advance. And this is, I think, a major increment that gives you very clear with a large enough cohort of patients is the key here, and more than half of whom went through the initial follow up. And bear in mind, there's still continued follow up from these patients, right? To come, God willing, for as long as possible. And yeah, I think it's warranted to do more along these lines, but like to commit to this one therapy, as we've alluded to in the past, when there's all these other approaches now, you got to wonder how we're going to marshal our resources to best serve not just the unmet need, but these patients that are entering these trials. How many things are we going to try before we move with something that really is practical on all levels, not just like the proof of concept, but like the feasibility on the economic scale and like the level of functional improvement, et cetera, et cetera, et cetera. So, like, I don't think I have the expertise to appreciate the finer points here, but big picture, I love the work, I love the results and the value, but it gives me kind of a bad feeling about the big target from the day one, which is we're gonna be injecting cells in the heart, we're gonna be using cell-based patches, whatever. I feel, like, less enthusiastic about that lately.
Yeah, I don't know, maybe because you and I have both been in this game for so long and we've seen this in the development for so long. I'm still optimistic in part because I know this is an early phase trial, right? Like this is a proof of concept more than anything. And one thing to actually clarify, there were some arrhythmic events. They didn't actually call them arrhythmias. There were arrhythmic events in four patients actually. So they were not the big one, which is VFib, ventricular fibrillation, which would be a huge problem if you're introducing those patches into the ventricle and it's causing VFib. It was what they noted as ventricular tachycardia, so the heart rate went up, and also atrial fibrillation, which is again not the site of the transplant. So unclear if that's associated with the particular the patch. But yeah, I know what you're getting at. I think maybe part of your concern is stemming from that conversation that we had with Deepak Srivastava, which
Yes.
I think spooked us. It spooked both of us because this is the guy, and Deepak actually had a great commentary on this particular paper that just came out because this is one of the leaders in cardiac cell therapy and IPS cardiac biology who's not as worried about, of course, he's concerned about the science as always, as a good scientist should be, but I think his big takeaway was the economics, right? The economic feasibility of some of these cell therapies, and is it really going to cross the finish line because of those, third party considerations? That's I agree with you on that. I think that's a major consideration here, but I think from a scientific point of view, I think it's a really exciting proof of concept, and again, a big hallmark 20 years into the IPS world.
Yeah, great call. And yeah, I mean, 30 years almost into the whole idea and the promise being really tangible. So, definitely warrants NEJM publication. I'm really glad that came out, and Loved reading it. Moving on to a nature story that is, I don't know, kind of related. In terms of like insult, right? We talk about MI, it's this massive infarct, and it's the scar of which is the problem, right? In the surviving patient. So this is a kind of related story, talking about stress and inflammatory insult. Except in the hematopoietic compartment, right? And I like this intro for this article because it gives you some kind of scope in terms of the numbers of what kind of a job it is for the hematopoietic compartment to maintain homeostasis and also be responsive over the long term of a human lifespan, right? So just some numbers here. We're talking about billions of cells and a huge amount of turnover, but to put a finger on it, we're talking 3 million cells per second that we're generating in our hematopoietic compartment. And of those, about 50,000 to 200,000 in any one time, like you're walking around right now, there's around 50,000 to 200,000 hematopoietic stem cells that are contributing to all that cell turnover, 3 million per second, right? And as we age, of course, there's a decline in the function of not just the stem cells, but all their derivatives. And that comes with a decrease in the clonal diversity as you get these dominant kind of clonal derivatives, the so-called CHIP, clonal hematopoiesis of indeterminate potential. And that comes with this increased risk for malignancy, right? So there's a lot about that process and the balance, right? Because we get all these snapshots and we have all these reconstitution assays, but over the long term, how does blood, you know, maintain that responsiveness, mobilizing in response to stress by, while also, with the steady state, everything. How does it do that? And also, how does it do that in the face of all these insults, right? Well, uh, there's tremendous heterogeneity, right, within the hematopoietic compartment on the transcriptional, also epigenetic level, functional level, right? And a big part of that heterogeneity is the inflammatory profile of those cells, right? Given across the entire spectrum, there's pro and anti-inflammatory hematopoietic cells, but also on the receiving end, there's a lot of evidence to suggest that over the course of lifespan, that these hematopoietic compartments are shaped by that inflammatory stress and it alters their profile. So this was a study, as I said, in Nature from Stephanie Z's group, who's at the Princess Margaret Center, Cancer Center, is the University Health Network of Toronto. And the question here is how do hematopoietic stem progenitor cells respond to and adapt to inflammatory stress? And in order to ask and answer this question, they developed this clever, recursive, elaborate, but elegant uh, xenograft model where they take human cord blood, they xenograft it into these immunocompromised mice, and then they expose them to these inflammatory stressors, recover the cells, and then engraft again. It's like serial engraftment, except with a inflammatory stimulus in the middle. Um, and then they take the output of that and use single cell multiomics and other, reconstitution assay analysis to analyze these cells. And what the big upshot here was they identified this novel cell type that they termed this HSC inflammatory memory cell. Okay, so HSCIM. And this is a cell, as the name suggests, it maintains this imprint, a molecular imprint of previous inflammatory treatments that they provide to these mice, the graft-bearing mice. And that profile also coincided with increased our tendency toward quiescence and restrained hematopoietic output. And here, very excitingly, and this is in terms of like clinical impact and relevance, this was, I think, a major story, probably why it warranted uh, Nature. They looked in adult and pediatric samples across many conditions: COVID-19 recovery, sickle cell, uh, as well as aging patients, patients with clonal hematopoiesis. And they showed that this HSCIM program was enriched in their blood samples, which in one fell swoop validated the xenograft, showing that there was this agnostic kind of concordance. And there again showed from like a diagnostic level how this could be a surrogate measure for these kind of disease manifestations, or perhaps the extent of disease or prognosis, using these HSCIMs as kind of a surrogate diagnostic measure. And then they did some more mechanistic analysis of these HSCIMs and showed that not only is it the profile of the HSCs, but their differentiated progeny maintain this pro-inflammatory transcriptional program, which there, right there, is a major key in terms of how this may have disease implications by skewing the entire hematopoietic compartment downstream of these inflammatory stimuli to a pro-inflammatory phenotype, kind of like snowball rolling downhill. And then here's the real kicker from a diagnostic kind of predictive standpoint. They show that the HSCIM program enrichment in circulating blood, so just a liquid biopsy blood sample from these patients with no mobilization, if you look at that and see a HSCIM program, you can see an association with heightened risk score for all-cause mortality. So kinda creepy there, the level, and of course, we're not talking about, like, diagnostic precision, like a one-to-one correlation or anything here, but, like, I wouldn't be surprised if someone is selling this. You get all these longevity experts talking on a blood test now that is gonna give you an association of your HSCIMs and inflammatory, not only as a measure of, like, how much inflammatory insult you've seen in your life, but also a predictor of all-cause mortality for you. Whoa! So, I mean, that maybe is pushing a little bit fringe in terms of the clinical import. But from a diagnostic standpoint here, the fact that they see this in not just age patients, but pediatric patients with like sickle cell, I think that this is maybe a new ball game in terms of like a novel cell type in the hematopoietic compartment, and not just one of these fringe cell types that exists within the spectrum, nuance, a little bit of that, like a brand new cell type that's associated with all cause mortality. Like I said, Arun, whoa.
Well, as somebody whose entire family has recently dealt with some inflammatory insults in our lungs, kids getting sick from daycare, pneumonia, all this kind of stuff, this is a bit scary to me. Maybe we should be profiling our own HSCIMs. And newsflash, that's why I wasn't on the roundup a couple weeks ago. Just had to deal with some sick kids. Um, but hey, I think this is very interesting as a diagnostic. I think the lifespan approach is perhaps a bit of a stretch at this point. But I think this is very interesting. The fact there is some memory associated in these HSCIMs for these inflammatory insults. And I think the other thing I really liked about this was how they broadly generalized this to multiple inflammatory insults, COVID, other forms of disease, etc. I don't think I searched for pneumonia. I didn't see anything in there, um, but yeah, I think it's definitely a very interesting topic, and something that I think, just like Chip, is going to have some diagnostic or biomarker value down the road. I think we're just kind of, at the tip of the iceberg here.
I mean, this is one of those things in life for me where it's like, do you even want to know? There's all I'm haunted by, yeah, COVID. I got COVID, and at the time, like, yeah, I'm robust, and I kind of shrugged it off. It was no big deal, but I did get sick, and like, I know a lot of people got really sick. And if I could give them a test right now, it's kind of like the Huntington thing. If I could give you a test right now that told you how much damage was done, I don't know. I'd be like, keep it. But on the flip side of that, not all knowledge is good, first of all, especially if you can intervene, right? And of course, then having knowledge of how banged up you are, you can live a more careful life. So no matter what. But from another standpoint here, what I always think of when I get these kind of diagnostic type studies is the theranostic extension there, right? Like in this case, if you have a kind of two birds approach where you can both identify here in case the HSCIM, and if that HSCIM is like the bad actor, then maybe you ablate it, right? And I feel like there's maybe a second half of this story, which is if you can modulate, if you can somehow find a technical way to specifically label and manipulate these cells, like what does ablation of these HSCIMs do? Is it positive? Is it negative? Do they have this kind of underappreciated this necessary value in inflammatory memory that's positive, or is this more like the senescent cell idea where we should get rid of them? And by that token, I mean senescent cells are no bad. That's the hook. So you got to wonder if maybe it's not just a matter of seeing how screwed you are, but maybe taking your future into your own hands by ablating these damn inflammatory HSCs.
Yeah, it's a good point. I think, one thing to consider it as a diagnostic, but like you said, as a theranostic approach, I think that could be really powerful. And I think there's a number of these in modern medicine that have really, bared fruit. The one that comes to mind is, of course, the test for BRCA, BRCA2, which, initially was a diagnostic and a predictor of breast cancer, right? But then there's downstream theranostic, downstream, tangible things that women can do to act on that test result, right? Mastectomy is being a big one, for example. So yeah, maybe that's the inevitable next step here, but I think there's a lot of work that still has to happen before we get there for sure. Um, Moving on to something that's, perhaps maybe a little more familiar to those of us who listen to the show regularly, and that's, of course, organoids. We talk about organoids all the time here on the show. This is a stem cell paper coming from China. First author Yu Gong, last author here is Jiawen Li from the Department of, importantly, ophthalmology. You'll understand why that's important in a second here. From Chongqing Medical University in Chongqing, China. So this is a very cool translationally oriented study using human retinal organoids to actually functionally bridge a transected optic nerve in a rat model. And this is you could think about the implications here for trauma, eye trauma. I've got a couple friends who are ophthalmologists. Unfortunately, they tell me horror stories about the things that they've seen, pun intended, I guess, in terms of eye trauma. And a lot of times it's, not reversible at all. So, you know, functional restoration of vision in adult mammals, it's still pretty much unattainable after you have like a really severe trauma, like in a bad car accident, complete transection of the optic nerve. It's not coming back. Um, Here they were trying to turn that on its head using some retinal organoid transplantation approaches. Pretty cool. So they're able to heterotopically transplant human retinal organoids into the transection cavity of the optic nerve in adult rats. So they induced the transection in the rats and then added the human retinal organoids to see if, very simply, if you can restore vision after the transplantation. And they were able to do that to some extent. So 1 month after transplantation, the treated animals actually exhibited partial recovery of visual function, including what, they characterized as a direct pupillary light reflex, so actually sensing light in some capacity, a visually evoked electrophysiological response, so the cells are electrophysiologically active and the circuit is active as well. Um, some visually guided behaviors, they're apparently figuring out where to go. And these, improvements in vision, quote unquote, were sustained for at least a few months, 3 months. And then they did deeper analyses, of course, as you always should, um, using some neuroanatomical tracing, three-dimensional imaging. And they're able to find that indeed there was a bridge, there was a structured host graft coupling that was happening, that was actually spanning the transaction gap. There's functional engagement of the graft, and it was further supported by a couple of inhibition studies that they actually did, some chemical and also genetic inhibition of the visual circuit, which actually led to an attenuation of the visual function. So, once they started seeing after the transplant, after doing the inhibition, they no longer saw, right? So it's I think a really cool proof of concept here that's showing that you might be able to use retinal organoids as a heterotopic transplantation approach to create almost like a relay-like pathway to support partial improvement of visualization, right? Partial improvement of sight, right? Restoration of the signal transmission across a completely transected optic nerve. That's what they were able to see. And I think it maybe gives some hope, right? It gives some hope and maybe in a conceptual framework for maybe repairing these severe optic nerve injuries for right now. There's not really an option to do so. So I think a very far-fetched kind of very translation-oriented approach, but one that I think is really honorable and I think, is medically very, very badly needed.
Yes. I mean, you said far-fetched there, which I think is you kind of amended, but that's what jumped to me. Not far-fetched isn't the right word, I guess. It's more like, huh, because you hear about the eye, and it's the RPE, and, we've seen this few papers talking about, rods and cones. But the transectional optic nerve, that's kinda out of left field for me. I'm not aware of the scope of that injury, so I learned a lot, the scope of that need, I mean, I learned a lot. But again, the is like, "Oh, I wouldn't have thought." Here, it seems crude isn't the approach because the analysis here is so sophisticated, and the result is so eye-popping, forgive me. But the mechanism, right? Cellular mechanism, it's like you're kinda like, "Huh?" And, I love a hack, so which is why I like this story, but it's just not what I would've drawn up, I guess, and, that's where I guess the next, questions will come is, like, what's the degree of the cellular contribution? Are there off-target cells in these organoids? They look very clean from what I saw. They did some single-cell seq, and the profiles were tight and not a lot of off-target cells, but, you've worked with organoids. There's all kinds of stuff in there. So from a practicality level, you wonder about, like, variability, I guess, between preps and whether or not this is something you worry about the safety profile in an immune privileged space where you're injecting cells that are not well characterized. But that's details because this is like I go back to the beginning. This is a hack where it works. I mean, that's the thing about this. It's so impressive. You look at all the readouts and like not just the electro stuff, but the mice are finding their way, right? So pretty dope. And we don't talk enough about the eye realize a forum or a a platform for realization of the great promise of cell therapy, and this is really it, right? This is like I was blind and now I see. So some real miracle type stuff here. Very cool on the results. It's definitely some kinks to iron out from a cellular mechanism standpoint, but very cool, very promising, clinically relevant, has all of it.
Yeah, and you could think about the parallels to, say, spinal cord injury, right? I mean, perhaps that's a bigger beast to tackle as opposed to transection of the optic nerve. And I'm not trying to say that this is some massive unmet medical need, just to clarify. I mean, because it's a select trauma that happens in a unique situation, like car accident, or I don't know. You can imagine the scenarios, right? I don't have to visualize it for you. Um, but I think it's as a proof of concept, it's very exciting. It's very cool because there's also a very clear functional outcome. And I think that's what we like to see here is there's an improvement in the functionality and the behavior of these animals. So I'm excited to see the next steps here.
Yeah, I can't wait. And I mean, you said that's a great comp, but that's kind of what I was getting at. I mean, if you recall, all the stem cord stuff, spinal cord stuff, they go in on the purity, right? And that Geron trial from forever ago, that was suspended based on these off-target cell types and the cysts that form from them. So purity is a thing. Great comp though, and a similar outcome in terms of like a vivid demonstration of the win and from a functional standpoint, that's a great segue for my story here, and it's a bit of a callback to what you mentioned about Deepak, which has been haunting us both. That interview and what he said about the economy of these hematopoietic stem progenitor cell engineering therapies. Um, because yeah, talk about a functional outcome that really demonstrates the win. These are patients who are struggling, suffering, and they walk out of the clinic cured, right? Not immediately, but this has been clearly demonstrated now that there's no argument against using this therapy except the the fact that it's not necessarily practical at scale and like the payer format hasn't been figured out yet, but I have hopes that it will. A big part of the problem there is the technical obstacles, you know. CRISPR wins the Nobel Prize and suddenly everyone assumes like you can target anything. But there's a lot of limitations and technical realities, right? Like, for example, you cannot target gametes, right? Because the genome is tightly compacted. Another example, and this is where we're going with this story, is that the idea of homologous directed recombination for repairing a genomic sequence that's confined to cells that are cycling, that are in SG2 cell cycle phases, right? So you can't really use HDR based editing in quiescent or even slowly cycling non-proliferative or slowly proliferative progenitor cells, right? And that's just one part of it. The other thing is that, of course, as we discuss a lot more, I think that's no one talks about the cycling element for HDR. But also, like, there's this whole other, which we know about, mechanism of recombination, which is the non-homologous end joining, right? Which is kind of destructive, leads to these indels, right? And there's the divergent outcomes. You get HDR at a much lesser frequency than you get the non-homologous end joining. So, like, yeah, in any one targeting event for a patient, you're going to get a lot of non incorrect targeting or inefficient or ineffective targeting, right? So there have been a lot of improvements. There's been a lot of proof of concepts, optimization, but the clinical application of homologous directed repair is way behind, say, other therapies that are more hacks that like disrupt the non-coding sequence or other means that are more robust, right? And this is magnified by the problem that every time you do one of these HDR events, it's not just that you're inefficient, but you could also be introducing this genotoxic risk, right? And there's a growing body of evidence now that's showing that there's large deletions, chromosomal translocations, or insertions of like either the entire or fragments of that HDR template that you're using to direct the repair. You find them either at the site or all over the place, right? So it's happens relatively frequently. So you're getting these heterogeneous populations of targeted cells that are not only not all cured, but some of them are like now maybe malignant potentially. It's risky, right? So not just like the economy here, but even just the practicality in terms of the wholesale application of these therapy and across the entire scope of unmet need. I don't know if we're there yet from a safety profile for HDR at least, right? So, this is a story here, which is the technical, approach from Luigi Naldini's group, who's at the San Raffaele Scientific Institute in Milan. And, Naldini, he's a legend for lentiviral therapy, right? He created a lot of the early uh, vectors and approaches. And I think what's great about the general approach is the sophistication and multiplicity of regulatory factors that are embedded in these cassettes that makes them practical from a clinical scale. And this is another example of that'll be widely used. It's kind of a variety of approaches that are called they're called selection by means of artificial transcriptional activators. So the acronym there is SMART, which is, I don't know, maybe in Italian that comes out to SMART, but it's so-so in the acronym. I love an acronym, and that one's an 8 out of 10. But the application is where it's at, right? And these are there. It's look at the paper in terms of the technical elements, but they're essentially these like regulatory kind of logic gating on a transgenic delivery scale. And this is, I think, in keeping with a lot of the approaches now that I wouldn't call it synthetic biology, but there's all this complex vector delivery that allows a very finely tuned delivery and regulation of exogenous factors in the genome, and this is one of them. And the key here is it allows you to deliver this HDR event, but then to enrich for exclusively the cells, or almost exclusively for the cells that actually have the correct integrant, right? And here they show, again, look at the technical elements, but here they show that they can enrich to 80 to 100% purity with these selectors, which are these transient selectors that then get diluted out once in the correct cells. Um, and not only did they get 100% purity of those guys, but of course, by extension, all the off-target edits were completely depleted, right? And then they use xenotransplantation assay to validate this in the immunodeficient mice. They All of the framework is in this clinically compliant manufacturing SOPs, and the selectors all again GMP compliant. So ready for prime time. I think Naldini's hitting the ground running here with these vectors that support like safe harbor integration for transgene overexpression or HDR gene correction. So something to, I think, plug into the pipeline here. I don't know about the economy element in terms of scaling up these therapies, but the safety profile, I think, that's necessary to keep them pristine from an optic standpoint, and from like just for these patients, right? We don't want to be introducing, and there's a kind of I don't want to say a dark history, that there's been some cautionary tales of therapies introducing malignancy. So I think this is a real key prerequisite to scaling this up. And as is typically his custom, Naldini introducing vectors that are going to be very widely cited, I'm sure available on AdGene. So anyone looking for a selective and specific expression in this context, have at it.
Yeah, shout out to Dr. Naldini, one of the real pioneers in developing these tools for the community. And this is particularly useful. And I liked what you got to at the end there about how these are clinically compliant off the bat. So really easily adaptable to GMP workflows. And that's what you got to do for various downstream HSC-based therapies. So I think it's a very nice tool for the toolkit, and I think a perfect fit for nature biotechnology.
Yes, I can remember one of the first things I did in my postdoc was hack a Naldini lenti to make this reporter assays. People still asking me for that. So shout out to you, Luigi. You invented me, or at least got me going. And moving on to our interview, where we're gonna talk about a little bit of inflammatory action in the heart. So apropos, before we get there, though, a quick message from STEMCELL Technologies. If you're participating in a life science focused journal club, STEMCELL Technologies is inviting you to enter to win. $350 are equivalent toward refreshments to fuel your journal club discussions. Oh, this is so fun. You guys got to enter this. The contest runs year-round, and winners are selected at the end of November and May each year. Increase your chances of winning by getting other members of your journal club to enter. To enter the contest, visit www.stemcell.com/stemcelljournalclubcontest. I don't think it's really fair, Arun, because if I win this, then they're gonna say it's a fraud, so I can't even enter. And I want that. I want that prize. I mean, that's awesome. All right, guys. On today's episode, a special guest from the University of Oxford, where she is group leader, also co-director of the Oxford Organoid Hub, Dr. Filipa Simões. The Simões Laboratory is interested in dissecting the dynamic cellular crosstalk and spatial relationships occurring between macrophages and the regenerating cardiac niche. In her lab, they use cardiac organoids to understand fundamental principles of heart development and regeneration. They also combine a wide range of state-of-the-art technologies to dissect the spatiotemporal dynamics of cellular microenvironments, identify extracellular and intracellular signaling networks, and decipher how these converge to define macrophage identity, plasticity, and function. Filipa, thank you so much for joining us on the show today.
Thank you for having me. It's a pleasure.
The pleasure really is ours. This is subjects that are near and dear to our hearts. I'm coming at you with the puns today. My apologies in advance, but Arun loves cardiac, so do I. And hematopoiesis is, my first love. So yeah, as I said there in the jump, you're focused on wound healing pretty much in the heart, and the convergence of immune, hematopoietic, and other cell types. And while cell-based approaches for cardiac regeneration have long been a centerpiece of the show, every episode we're talking about the heart, feels like, in the more traditional cell-based therapy, if you could call that traditional. But we've recently highlighted strategies that are more focused on modulating the immune cellular environment within the infarct zone to mitigate loss of contractile function. So why don't we start with you giving us an overview of your lab's focus on macrophages and the relationship to cardiac wound healing?
Sure, thank you. Like you said, Dylan, we really are fascinated by this very fundamental question in biology, which is how do complex tissues develop and regenerate upon injury, and in particular, how the heart does this, both during development and regeneration. We have these progenitor cells that need to really follow a very specific and orchestrated differentiated path to give rise to specialized cells that then need to orchestrate and really talk to each other to form this really organized 3D structure and ultimately a functional organ, a functional pumping heart in the case that we are really studying. And the way we address this question, and we really tried to address this question in the lab, is by using two very complementary approaches. One that really uses these 2D and 3D stem cell systems, where we really study cell fate decisions in a kind of in vitro manner, but we very much ground this work on fundamental principles that we discover in vivo. So really the idea that these cells are talking to each other to really build a functional heart is true during development and during regeneration of the heart, when you get this beautiful organ being put together for the first time or being rebuilt. It's not enough that you make the different cell types that form the heart. It's not enough that you form cardiomyocytes or fibroblasts or epicardial cells. They really need to be organized in a 3D manner, talking to each other, where this signaling crosstalk is crucial from our viewpoint to really make a functional heart in the end. So we really try in the lab to make this cellular crosstalk experimentally tractable. And unifying paradigm that we use, like you very much alluded to, is really this idea of the macrophage niche interactions that are present both during development and during regeneration, and really use that concept to really try to dissect how the microenvironment first program these macrophages. They are incredibly plastic cells that are present both as the heart forms, but obviously upon injury, and I'll tell you a little bit more about that later on. But really then we use these principles to then understand how then these macrophages in turn also shape tissue outcome and rebuild the microenvironment itself. And in vivo work for us is really based on the zebrafish as a model. So zebrafish, obviously in development, is this incredibly powerful animal model where you can use a lot of imaging technologies and obviously genomic technologies to really dissect the principles of heart development. But perhaps in the terms of regeneration is one of the most well-studied models, really, because they're this champion of regeneration. They can really regenerate many organs, including, of course, the heart. So macrophages play an integral part here. We know quite a bit about macrophages in the regenerative or reparative context. They are amongst the first cells to reach the heart after injury. They have this more pro-inflammatory phenotype to start with, where they go there to the injury site, they really clear up the dead myocytes, the apoptotic neutrophils, and then this kind of pro-inflammatory wave is more superseded by a pro-reparative or anti-inflammatory wave of monocyte-derived macrophages, as you know, and these are in a way really good at suppressing inflammation, which is important. But in a way, they do it too well, and really this response leads to the formation of a fibrotic scar. And this is true in a regenerative model and in a non-regenerative model, which is obviously adult mammals, including humans. The difference here is that with the zebrafish model, the nature of this scar is transient rather than permanent, like it is in humans. So we can really study how macrophages are influencing these different stages upon injury, including scar deposition, but also scar removal at the same time that new cardiac muscle cells and new blood vessels are formed to really restore the heart as a functional organ.
Yeah, I mean, I've told the story a little bit before about how the zebrafish cardiac regeneration phenotype was one of the big reasons as to why I became a cardiac biologist in the first place. I was an undergrad at Duke University, and of course Ken Poss was there for a number of years, and I was just, so enamored by the work that he did on cardiac regeneration for many years. It's such a powerful system, like you alluded to. It's genetically malleable, it's transparent, easy to image. Talk a little bit about the zebrafish and just the power of the system and how you first got involved in the system in the first place.
So I first got enamored, like you said, by the zebrafish really when I started my DPhil here in Oxford, working in the lab of Roger Patient, which is very well known for his work in the hematopoietic field, but he also had a smaller part of the lab working in heart development. Was very much what I studied for my DPhil, where I was very interested in understanding how early on during development these cardiac progenitors get specified in this anterior lateral plate mesoderm. And like you said, the power of the zebrafish is really that development happens very quickly. So within 24 hours, you have this beautiful beating heart that, like you said, we have capacity to generate many transgenic lines that allow you to follow processes in vivo in real time. So really the power of seeing how these things are happening in a vertebrate model. Of course, more than 80% of the genes are conserved, and sometimes it's not that the same exact player is conserved to humans, but usually the same molecular pathway is conserved. So that really gives us a handle on understanding fundamental principles of development in this case, as I was studying. And of course, we used to have morpholinos as our bread and butter, but then we moved on to more kind of genetic approaches. And of course, with CRISPR, we really are able to generate many mutants either in a transient manner or in a stable line manner that really allow you to then have a understanding at the functional level and mechanistical level of how these gene networks are really established to really form what is fundamentally a rudimentary heart to start with, but then that very much the same genes and molecular and cellular processes are in place as it happens in our own heart.
Yes, I mean, that is the conservation of those molecular cellular processes is the key, right? And I mean, the tool set for zebrafish, as you just described, is extensive and has become more and more sophisticated and so amenable to the live imaging and, in on water, so to speak. So you can really see these, beautifully orchestrated, you know, cellular ballets right before your eyes, and with these markers. It's really a beautiful system. But as I said, the conservation is the key. And you alluded to the capacity to retain regeneration into adulthood that is not a facet of adult cardiac regeneration or repair, we should call it.
Hmm.
Um, but, it wasn't too long ago, I don't know, maybe a decade or so ago, that there was this real, you know, revolution, bombshell in the field, which gets back to your DevCell paper. So I want to talk about this DevCell paper from a couple years back that interrogated epicardial gene expression in the regenerating zebrafish heart, right? And as I just described, such a beautiful system. So the story was very rich visually, and the mechanism was elaborate. But for me, like conceptually, I was kind of scratching my head because as I was building to the story was that in zebrafish, and correct anything that I get wrong here, um, but there's distinct molecular cellular programs that are deployed in the epicardium during the embryonic, building cardiogenic phase versus this adult reparative regenerative phase, like completely different mechanism. And why I was struck by this is because I was really invested in this hypothesis that emerged about a decade ago from the neonatal apical resection or cryo, whatever, in the mouse heart neonatal phase, and it would repair. And everyone was like, not everyone, a lot of people were like, well, there it is. Like, there is this capacity for repair in the mammalian heart. It's just, it's only in the neonatal phase or it needs to be reactivated. And there was a lot of work focused on that specific repair and whether or not you could transfer that mechanism into the adult mammalian heart. And I would say to modest, if any, success to date.
Mhm.
So, I mean, speak to that.
Mhm.
In your view, is that a hypothesis that you think is still viable? This idea of transferring a neonatal or even non-extra species repair process into the mammalian heart, or do you think that's really fundamentally limited, and it's more, as you kind of are going for, this kind of immunomodulatory, kind of mitigating the damage of the repair as opposed to actually regenerating? Like, what's your take on that species transfer or neonatal
Mhm.
endowment, whatever you want to call it?
Yeah, it's a super interesting topic, and I think what you said is really absolutely valid still today. I think for a lot of the processes that happen, at least during regeneration, and to a certain extent in a reparative kind of model, it's a recapitulation to at some point what's happening in development. So when the organ forms in the first place. So a lot of it is a redeployment of this same blueprint, which is why it's so fascinating to study these very fundamental principles in the embryo. So we can really try to recapitulate some of it in the adult setting. But of course, there are very big differences. In development, everything happens orchestrated in a certain time point and in a kind of slower time scale in a way where, like you described earlier beautifully, this kind of orchestrated dance of cells is really happening in a beautiful way. In a regenerative or upon injury, everything has to happen super quickly, right? So it's almost like the first thing that needs to happen is really, oh, we cannot die. We need to do something immediately here.
Mm.
And of course, with that, we might overdo it, right? And that's kind of survival. Again, probably evolution will have a lot to say here. But so going back to the study that you described, that was a joint study with Michael Weinberger in Paul Riley's lab and Tatiana Soka-Spengler's lab, what we really found out is that even if some of the gene program or the genes that are reactivated upon injury might be the same, what we really found that was quite interesting in our perspective was that the regulatory networks that are actually activating the embryonic gene are not necessarily the same as the ones that are being activated upon injury. And that might be just because, again, time-wise is very different, or things that are there in the adult might be in a more steady state, right? The heart before injury is in a homeostatic state. So things are more poised, are resting, are kind of keeping everything in check. So maybe the easiest thing to do is almost to take out the brake and let everything go and repair very quickly. So again, the organism doesn't die. But also it's an incredibly complex system, of course, and that's why we are so excited about it and to continue to work on it, because it's not enough to modulate the immune system. It's not enough to control the formation or removal of a scar. You still need to make new cardiac muscle cells. You need still to make new blood vessels. You still need to remove all those fibroblasts that got activated, all that collagen that got deposited and hyperlinked and so on. So it's not, I don't think that it's going to be one approach that will really nail this down. But the good news is that, again, technology-wise, we're advancing so much at with work that everyone is really contributing to. So I'm hope. And also the other thing is we really learn from other organs and other systems where very similar principles apply, again, with the participation of the immune system, with the fibrotic scarring approach, with the need for new tissue to be built and so on. So there's many parallels that I think can benefit us in terms of thinking about what approach to take moving forward. But it is highly complex, as you would expect, yes.
And I think, one of those other complexities, and it's something that we've talked a little bit about on the show recently, is the importance of, mechanical load on the development of the heart. It's such a biomechanically dynamic organ. In fact, you probably saw that science paper very recently that was talking about how that's a really important reason as to why the heart probably doesn't have tumors, right? It doesn't get cancer, which is an amazing paper to see. Talk a little bit about that, the importance of, mechanobiology and influencing some of these processes. Is this something that you guys are studying?
Yeah, so I think this is a super exciting field that is really now, I think, booming even more into the field of developmental biology. And even one principle that I'm actually quite excited about is to link. So we work in a very genomics kind of viewpoint. So we study a lot of these principles from a genomics perspective. But what we're now starting to do, working with colleagues like Tim Saunders, which are truly physicists, mechanobiologists, is really understanding what is the connection between the genomics and the mechanobiology there. Like you said, obviously the heart, from the time it's really it starts pumping so early, and then is a 24/7 job until until we're gone. So the load there is incredible. So of course that needs to be really factored in. And I think so far people see it in isolation. There's people that will really study these principles or these processes, really focusing on the mechanobiology aspect. Others very much on the more like cell biology or transcriptomics or so on. But I think we really need to blend this and unify it, because there'll be feedback loops here happening, and where the mechanical aspect really influences cell fate decisions, and in turn what a cell becomes then informs aspects of the mechanobiology. There's colleagues that are doing really beautiful work in this field, in the heart and beyond. And I think that's going to be something that will be quite exciting moving forward. And this paper that you just mentioned is truly fascinating. And the heart is one of, if not the only organ that really could. It's very rare that you really can see any cancer. So this is quite a cool suggestion why that is the case. Yeah.
Yeah, and it just so happens right next door to you, or in collaboration, you're actually
Yeah,
working very
yeah,
closely with a
yeah.
mechanobiologist who I actually, trained with, and Dr. Christopher Topfer at Oxford as well. So I think you're at a great place that supports this kind of work.
Hmm.
The colleagues are certainly there for it. I mean, you've got an amazing group in your own right, but also the network there at Oxford, which we're going to come around to in a minute in the close. But just speaking to that work, this whole idea, as we've been kind of dancing around the complexity of heart repair and all these factors, it's an engineering problem, it's an immune problem, it's maybe a cell regenerative therapeutic problem. And you really need all hands on deck, so to speak, to address it. But I think that this idea, as we've talked about in the show recently, of engineering immune environments rather than just focusing on replacing lost cardiomyocytes is really coming to the fore as maybe the best first approach. So can you just talk about like what you would say, not even necessarily low-hanging fruit, but what do you think is the most critical problem or the first best target for immune modulation, engineering an immune environment in the heart, and whether or not it's something that could be generalized across the board, or but more specifically just in the heart. Like, what's the thing that you think is the biggest problem that you're working on right now and has the greatest chance of changing the game?
Yeah, that's a really great question. Like we alluded earlier, it's obviously a very complex problem. But I do believe that one of the ways that could really target a lot of these principles or processes that need to be in place is really via the immune system, in particular macrophages, right? So one particular angle that we're very invested in is really this idea of the microenvironment and how the microenvironment really shapes macrophage identity and ultimately function. So in my postdoctoral work, one of the really interesting findings we realized is that macrophage. So we, as a dogma in the field, we always thought that the scar that is produced upon injury in the heart is really produced exclusively by fibroblasts that get activated by macrophages and then become these myofibroblasts that really are producing all this collagen that makes up this fibrotic scar. What we found both in zebrafish and in the mouse, so being very much an evolutionarily conserved process, is that macrophages can in fact directly deposit this scar upon injury. So they not only upregulate all these collagen molecules upon injury, they are actually able, we've proved that by, or we tested that by adoptive cell transfer experiments, where we're able to show that collagen is really deposited into the scar directly from macrophages. And what we noticed in the zebrafish is that the location of this macrophage deposited collagen was very much in co-localizing with this outer layer of the heart called the epicardium, which is really this first cell layer that really almost regenerates and responds very, very early on. So we kind of started wondering, why are these macrophages that are clearly having a profibrotic phenotype, they're really contributing to scar formation, why are they located just in the epicardium and not elsewhere? Which really kind of gives this idea of trying to understand how these gene programs are being set up in these macrophages, really depending on their location. So this could really give us a handle on understanding cell-cell communication that is very much anchored by spatial configuration, spatial position, and how this chit-chat that is happening between the subpopulation of macrophages that is very profibrotic or is very pro-regenerative is different. So what is the cell that this macrophage A is talking to, and what are they talking about that is different from what's happening with the neighboring ones? And so this could really give you a much more molecular grounded insight on how to modulate and perhaps be envisaging how to modulate their post-injury response from a more profibrotic to a more pro-regenerative. So in fact, we have currently as a preprint and a review, a study that is really pinpointing this really very fundamental question of spatial, so cardiac immune interactions, and how this is really affecting the different macrophage identity and ultimately tissue outcomes. So we found this particular subpopulation of fibroblast that expresses IL-34 that seems to be communicating with a particular tissue resident population of macrophages and activating this transcription factor called EGR1, which seems to be really kind of working almost as a switch, and in a wild type situation really helping the regenerative process to progress towards regeneration. But when we really take that kind of promoter out and we really functionally manipulate this pathway, what we see is that these macrophages stay in this sustained inflammatory state, which really then inhibits the regenerative process and really leads to a fibrotic scar. So this is just an example of, by looking at how these particular interacting fibroblasts are talking to these macrophages, we really got a handle on the molecular mechanism, how you could really play with this switch to really either push fibrosis or push regeneration. And of course, what you want ultimately is to push regeneration and inhibit fibrosis. So I think this could be a more molecular, insightful way of modulating this macrophage response.
Yeah, it's really looking at two sides of the coin, yin and yang, whatever analogy you want to make there. And like Dalen alluded to, you're of course at Oxford, which is an amazing institute to actually get to the bottom of some of these fundamental biological questions in the heart. I mean, Oxford, as a lot of folks know, is the oldest university in the English-speaking world. I mean, there's a story that we love about Oxford that's, meant to highlight the need for long-term stewardship, right? The story goes that when it was discovered, the beetles apparently had destroyed the giant oak beams of the dining hall. And apparently the college forester replied that the founders had planted an oak grove for that explicit eventuality 400 years prior. So there's a huge history there, in this university. But you're part of course, a much newer venture at Oxford, the Institute of Developmental and Regenerative Medicine, which has the mission of pioneering new drugs and therapeutic strategies to combat chronic illnesses. And of course, organoids are a big part of this as well. So what makes this IDRM exceptional, and why do you love working there?
Yes, I mean, Oxford is incredibly special for the reasons you mentioned. It's really about the colleagues and the people that we interact with. So I feel very fortunate that I look up to my colleagues and I get inspired by their work, and really motivates me to do better science. So I think a lot of it is that, and it's very much so. The IDRM it's a quite special. It's like you said, it's very recent. The institute opened in 2022, and what's really exciting about the IDRM is that everyone in that building is very interested in development and regeneration, but across different organs. So we have a neurosciences aspect, immunology, and cardiovascular. So one of the things, of course, it's really nice to be surrounded by colleagues that are studying the heart in a very complementary way. But to me, one of the things that excites me the most about the Institute is, in fact, what colleagues in other floors are doing. So what colleagues in the immunology floor and the neurosciences floor are doing, because often, and we kind of touched on that earlier, often we face the same challenges. We study very similar processes. We're just looking at the different organs. So I like to get inspired by things that colleagues are doing in different fields to really then transpose that and adapt that to the heart. So that's one of the things I like the most. And of course, there's a big push on technology around here. So again, not only on the in vitro space, but the in vivo space, I think we are fortunate that if you think about something that you want to do, you knock on the door of a colleague around Oxford, either, in any institute, not only at the IDRM. And I've been here for a while now and never faced anyone that said no to a crazy idea that I've proposed or a new collaboration that I would love to initiate, and so on. So it's this highly collaborative environment with people that are better than you around you, that really push your work to the frontier. And I think this is one of the things that I really appreciate the most. And then just touching to the college kind of illusion that you just mentioned earlier, that is something that is also quite special here, because so in your institute, in a way, you're surrounded by people that, I think very much like you, or at least have very similar background. But when you go to the college environment, you're really exposed to these incredible people that are doing things that are so different from what you're doing. So this, for example, today I just had lunch with an historian, a mathematician, and a physicist, and we're talking about, so anything, nothing to do with the heart, but with all these things that in a way really make you, again, think a bit further and out of the box. And this is something that is, again, quite special about this place. That and the fact that the city is very tiny, right? We are probably just over 120,000 people here. We have probably, I would say, 3 main campuses that if you have a bike, which most people do, you can reach within 10 minutes. So really, there's no barriers really to be reaching out to all these colleagues around here and really take your science wherever you want to take it.
Wow, that's a really inspiring rendering. My takeaway there is that there's nothing better than being awed every day, even within your own field, like of science, and even a little outside of it, maybe not cardiac, but something related. But even like out to like philosophy and history. I mean, that must be so cool. I'm jealous, but a
Come
great
and
place to
visit.
do it. You don't have to ask me twice. I'll book a flight right now. The history out there is great. And walking the halls shared by all the real premier thinkers of our time, some of them at least, many of them. Um, that's a great capstone to a really fun conversation. Before we let you go, though, we have a couple of peripheral questions. First, if you weren't over there awed every day, doing your science amongst the thought leaders in all fields, what do you think you would be doing as a professional career?
Yeah, I think I consider myself extremely lucky because I absolutely love what I do and being a scientist. So it's hard to imagine doing something else. But if I would have to pick a different career, I'm a very visual person and I love design and architecture. So probably would be something along those lines.
Hmm.
I ended up, marrying an architect, so in a way that gives me that complementary side.
Hmm.
But yeah, probably something more on the graphics kind of aspect.
Well, there's nothing like the heart to lie at the convergence of both art and, engineering. So,
Yeah.
apropos, finally, a kernel of advice for, I don't know, professional, non-professional, for trainees, for anybody, for me. I could use some advice. Filipa,
I
thumbs
think
up.
advice is always a tricky one because we all have survival bias, right? So I think I'm very open to talk about what worked for me just because I think it's very important to put the message out there. There's more than one way to do this, right? So for example, I had my children very early early in my career. I had my daughter straight after my PhD. And I think it's important to say that you can do it whatever way you want.
Hmm.
You don't need to. I mean, these days more than ever, I think you really should. If you want to do something, really go for it. So don't be afraid to fail. I tell my group to fail hard and often and early, because that's the only way. Really, be comfortable being outside of your comfort zone because that's the only way we can go there. At least this is kind of my view. But really, kind of this idea of you really need to have a reason to wake up in the morning and really be excited about what you're doing. I think there's I don't know, there is another way, but for me, I really need to be excited about my next question. I have this urgency to know. I want to know what's there, what's the next thing that we're looking at. And if you don't have that spark, I think it's harder to do what we do. But yeah, just keep going, keep pushing, and enjoy the ride. Otherwise, there's many other things we can do for sure.
Yeah, that's very meta, because it's a piece of advice, but also saying that it's not the only way. And my favorite part of that, I'm gonna put it on a t-shirt: fail hard, because that is the first thing you got to do almost. I don't think anyone's just succeeded right off the bat, and if they did, I hate them. Anyway, Filipa, thank you so much for sharing that advice and all your insights, and the invitation to visit Oxford, which is the most exciting part for me.
Please
Um,
do come.
I'm assuming that doesn't stand for all our listeners, but maybe it does. I mean, go have a visit. Uh, Filipa will host you, although you gotta get your own hotel. All right. Thanks again, and really looking forward to your next piece of work.
Thank you. It's been a pleasure. Thank you.
All right, you guys, that brings us to the end of our show today. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. If you enjoyed this episode, please feel free to share with a friend or a colleague or on social. You can also reach out to us on X at StemCellPodcast, on Blue Sky at StemCellPodcast.com, or via email at info@stemcellpodcast.com. There you can leave us some feedback, or you can suggest some guests, or heckle, whatever you want to do. Until the next time, which will be about 2 weeks from now, thank you so much for listening. We're also coming in hot with the ISSCR meeting in the coming weeks, which we are very, very excited for. Be sure to tune in for our on-the-ground daily episodes, which are also a treat for everyone involved, and it'll keep you up to date. Until we're in your ears again, thank you guys so much for listening.